Hi, today we're doing biomolecules in accordance to a BSc-MSc integrated course. We'll start from the very beginning. Biomolecules, right? This is made up of two terminologies. Bio, that means living organisms, like living, and molecule is the smallest unit that makes up an element, right? Therefore, when we talk about biomolecules, we say we're referring to the chemicals or the molecules present in a living organism. And here we come across this term cellular pool, right? This is basically the sum total of all the different biomolecules, the compounds, the ions that are present in a cell. We've come across terms such as gene pool, right? That means the sum total of all the genes that are present in a population. Likewise, cellular pool. And we're also aware how carbon is the element of life. Why is that called so? Because every compound you find has a carbon chain, right? And any group is attached to that carbon. So the element that all the chemistry of living organism is being organized around is carbon, right? So therefore it's very versatile. It's forming proteins, lipids, carbohydrates, right? All of that, and it's the most predominant element of life. Okay, life itself is carbon. It's compounds of carbon, right? It's also important to note what an element is. Basically a pure substance, right? It is only one kind of atom. Now you might ask, what do we really mean when we say one kind of atom? We're aware that atoms are made up of protons in the nucleus, right? And the way we organize it in the periodic table also has something to do with that, right? The atomic number is basically the number of protons in its nucleus. Like carbon has 6 protons, oxygen has 8 protons, right? So when we say it's this element, it's going to, however number of atoms it has, all of them will have the same number of protons. So that's what we mean when we say it's one kind of atom. Moving on. So now we're clear what biomolecules are, what elements are, and how carbon is the most predominant element of life. And we're going to talk about this experiment or technique we use. Okay, it's called chemical analysis. What the aim is, we're detecting the various types of biomolecules that are present in a living tissue. So we'll take this tissue, we'll grind it with trichloroacetic acid, right? Trichloro, chlorine, 3 molecules of chlorine, and acetic acid. We're going to filter the obtained slurry, right? It will be like a slurry with broken down elements, but a bit thick. Okay, when you filter it, you're going to get the filtrate. When you filter something, there's some of it here, and then there's some of it here, right? So this is the filtrate, right? This is the filtrate. This is the retentate. So logically, something that is soluble, right? We used an acid, so whatever is soluble in that acid will be able to come here, right? It will pass along with that liquid. So here we have the filtrate, and this is the retentate. Retentate meaning this what it retains, right? Retention किसका हो रहा है? So the one that is acid soluble is known as micromolecules or biomicromolecules. These are smaller, from the name you can get. This is going to be insoluble in acid, obviously. This is known as biomacromolecules. The difference, let's quickly summarize. The smaller one, less than 1,000 Daltons, right? And this will include monomers, right? Like amino acids, like nucleotides, nucleosides, and also simple sugars. Simple sugars are oligonucleotides or oligosaccharides or monosaccharides. So it's also important to note what is the difference between these terms we're using. We're familiar with this, right? Monosaccharides, oligosaccharides, polysaccharides. From the name you can get mono means it has one sugar unit, right? So this is like the simplest form. Examples being glucose, fructose and galactose, GFG. And these are highly soluble in water. These are acid soluble, also soluble in water. And oligo is basically few, it means few, so a small chain. While polysaccharide will have a number of sugar chains, right? So polysaccharide is obviously not a monomer and it is not acid soluble, right? So we've cleared that up. Also important to know what's the difference between nucleotides and nucleosides. You always hear them, right? But do we know the difference? They're both nucleic acids, or rather the building blocks of nucleic acids, right? So what is the difference in the tides and the sides? There's a very crisp difference here, right? Nucleoside has a nitrogenous base and it has a pentose sugar. Okay. So what it lacks is phosphate, right? It doesn't have a phosphate group. So the building blocks of your DNA, they are guanosine, adenosine, thymidine, right? So without the phosphate group, they're nucleosides. Okay, they make up nucleic acids and all that. While the one that has the phosphate is a nucleotide. This has base, sugar, and the phosphate group. So it can have 1, 2, or 3. So all that ATP, ADP, AMP, all of that come under nucleotides. So the difference is in the phosphate group being present, and therefore their function also differs. This is used in energy transfer, while those are the building blocks for nucleotides. Right, so nucleosides are actually the building blocks for nucleotides, of course. If it lacks something and you just add the phosphate group, it will present you with a nucleotide. So that's basically it. Let's quickly talk about this. This retintate, it's acid insoluble, so macromolecule with biomacromolecule. The molecular weight is more than 10,000 Daltons, and here we'll have complex carbs, we'll have nucleic acids, and we'll have lipids. Interesting thing about lipids is we can't strictly say it's a macromolecule because the molecular weight is actually 800 Daltons, but it also falls under the acid insoluble group. So these are polymers, right? So we have quite succinctly we know the differences between these two. Okay, now let's cover up some other important points here. Okay, we talked about we'll talk about the chemical basis of life. Okay, so basically, before we get into each and every element in detail, uh, let's just talk about the chemical basis of life. And the living organism were composed of about 25 chemical elements. Okay, the bulk is, we've already talked about carbon, right? We've already talked about carbon. There's hydrogen, there's oxygen, nitrogen. All of this makes up the bulk of it. Okay, then we also obviously we have calcium, we have phosphorus, we have potassium, sulfur, sodium, chlorine, magnesium, in this order itself. Okay, this makes up most of the body weight, and with lesser amount, this being 0.1 percentage of the body weight, and this being 65. So um, let's just name the chemical compounds. We've already mentioned them, right? There's carbs, there's protein, there's nucleic acids, and there's water. Okay, these are the compounds. You already know this is H2O, right? So hydrogen, oxygen. Carbs में क्या होता है? CHO, right? There's all the chains you see of glucose and others. You'll see there are compounds made up of groups of carbon and hydrogen, the double bonds or single bonds, all that. So these are the basic elements of those. Okay, proteins additionally also have nitrogen, sulfur, phosphorus. Nucleic acids have phosphorus, nitrogen. Okay, this will be pretty easy to remember. CHO though is the common part here, and additionally we also have phosphorus, nitrogen, and sulfur. Okay, we've already talked about what a biomolecule is, right? It's an organic compound. It's essential component, right? Okay, it will be good to note down here that biomolecule is organic compound. Sometimes we get confused about even this term, so it's a very simple thing to also remember. Organic is basically anything that has a CH backbone, like all of these, right? But water is inorganic. It does not have carbon in its backbone, right? So these organic compounds generally have covalent bonds, and anything that makes up life related molecule is usually this. And the ones that don't have carbon, right, they are inorganic. Okay, water, NaCl, all of that. So generally non-living sources, minerals and gases, they are inorganic. And inorganic ones are actually soluble in water, while organic ones, they're soluble in solvents, organic solvents. Like is dissolving with like, you can say. And yeah, however, carbon dioxide, it has a carbon in it, but it's also inorganic, right? It's um, uh, not to say that organic always is bonded with hydrogen. It always has carbon and usually a hydrogen. So carbon being the key here to knowing if it's organic or it's inorganic. Anyway, so biomolecule, right? They're organic compounds. They have covalent bonded backbones. These are the building blocks of cells. And all these biomolecules then participate in dynamic functions, right? Everything being carried out is being done by these organic elements inside a body. So we've already discussed macromolecules, right? What were the differences? Except the molecular weight, you can tell it's going to be small sized. It has a low molecular weight. This is large, high molecular. This is acid soluble, acid insoluble. Additionally, there are some more elements we can remember, such as gases, water, minerals, right? All of these are also micromolecules. And lipids, we've already talked about that. Proteins as well, they're polymers obviously. Okay, now I'll introduce a chart here for your reference. Okay, these are telling us what is the building block of a certain molecule, right? Protein has amino acids, DNA, deoxyribonucleotide, RNA, ribonucleotide. Monosaccharides make up polysaccharides, while lipids are made of glycerol and fatty acids. The major function, DNA, hereditary information, right? Protein synthesis, RNA. Storage form of energy, polysaccharides. Storage form of energy to meet long-term demands is lipids. And basic structure functioning is the responsibility of protein. Okay, so you can do match the following of these, mix it up, just to remember. Yeah. So like we talked, CHON, these make up 99% of the atoms in the human body actually. Interestingly, right? So let's do some salient features, some important features of biomolecules. Okay, point wise we'll go over them. Okay, we already talked about how most are carbon compounds, right? So carbon compounds, most of them. These organic molecules, they also have specific shapes and dimensions. And if all these organic molecules have the same backbone, what actually is determining the difference in them, right? Where is the difference in the chemical properties arising from? Okay, they're not all the same. So that is thanks to the functional groups that are attached, right? For example, we have hydroxyl groups, carboxyl groups, we have amino groups, right? We have carbonyl groups. We'll go into this in a bit, right? The oxyl, hydro, and carboxyl, like, what's the difference here? It's actually pretty simple. So, hydroxyl is OH group, right? Oxy and hydro. Carboxyl is COOH. Amino is NH2, and carbonyl is a C double bond oxygen. Okay. So this is basically what we mean when we say function groups. Aldehydes are carbonyl groups. Okay. The aldehyde groups belong to the carbonyl class. Obviously, carboxyl is carboxylic acids, amino acids is aminos, and hydroxyl is alcohol. This is the basic thing you should remember. Okay. So also OH, so this means it's polar molecule. Carboxyl means it's acidic, amino, basic, and carbonyl means it's reactive. Why? Because the double bonded, the carbon double bonded to the oxygen is actually a reactive site. Okay, so it's reactive. Next point, right? So most of the biomolecules are polyfunctional. Okay, you would not see one molecule just performing one function, right? It's reactive in different ways, and through reacting with different compounds, it is carrying out different functions. Okay, and it has also different groups at different points. So it's polyfunctional. It is carrying out multiple functions. Okay. Obviously, biomolecules are not 2D structures. They are 3D. They make up 3D organisms, so they are 3D, and they're asymmetric. Okay. And how is this 3D shape being maintained? Why does it not collapse, and why is it actually a 3D shape? It's because there are a lot of non-covalent bonds between the atoms in the molecule. So the 3D shape is maintained by a lot of non-covalent bonds between the atoms in the molecule. So again, we're discussing very important aspects. Okay, this sounds like it makes sense, but think about it. Okay, what do we mean by this sentence? So biomolecules, we know that they have specific 3D shapes. Okay, and while attachment to each other is covalent bonds, right? They are strong, chain-like bonds. Like if there's a protein, there's peptide bonds which are covalent. Okay, so that's a very strong bond. But the 3D structure is what? It's basically the folding, right? The folding. So this is actually rather weaker bonds. Okay, these are reversible too. Okay, these are weaker and reversible bonds, and we know them as non-covalent bonds. Okay. So this can be hydrogen bonds, it can be ionic bonds, the van der Waals forces, hydrophobic interactions. All those come under non-covalent bonds. Might be useful to actually note it down. Okay, hydrogen bonds, ionic bonds, van der Waals, and hydrophobic interactions. All of these are non-covalent. What is ionic? Meaning the plus minus side chains, right? The positively and negatively charged side chains, the interaction between them forms it's not a bond per se, but it gives it a shape, right? Then hydrogen would be a hydrogen atom that is bonded to an electronegative atom and another one. Okay, so it's basically a base pairing. The hydrogen bonds are the base pairings, right, of ATGC. Those are all hydrogen bonds. Okay, where the hydrogen is covalently bonded to an electronegative atom. Okay, so also if it's bonded to O or N, these are electronegative. That's a hydrogen bond. Van der Waals are weak attractions between atoms that are closely packed together. Okay, So when they're closely packed, there's temporary dipoles that form. That's basically it. And hydrophobic would be non-polar, non-polar regions are hydrophobic, right? So they'll start to cluster away from the water. Okay, and when all of them together are clustering away, okay, what would happen? It would form like a shape that's again folding. So this is the important part here. The backbone is covalent bonds, but these non-covalent interactions are going to hold the shape of uh, that compound, that biomolecule together. Okay, and that's why we can actually denature a molecule. We can actually make it lose its functioning. Why do they lose their function? Because there is a change in the shape, right? And how do you do that? How do you break them? Obviously external, you'll give them entropy, right? Heat. You can even change the pH. That's why pH is always so important. It holds the shape together and thus the function. Okay, that's important to remember. Also, I used another term that I think might have struck as a question, right? I talked about dipoles. What are dipoles? When did I mention it? When I was talking about van der Waals folds and how closely packed together atoms have these temporary dipoles, right? So, so this is actually a really interesting concept and such a key interaction that happens. So a dipole is basically if you have a molecule or a bond, right, between two, and in that same molecule the positive and the negative charges separate out to two ends. Okay, so in that same compound, what in the molecule, some of the atoms are electronegative. So you must have heard of electronegativity, right? It's the property that it pulls atoms, or it pulls electrons towards themselves very strongly. So these atoms pull it more strongly than any other would. So there's a separation. A good example is, let's talk about, well, this is an example of a permanent dipole, right? What happens here is, if we talk about H2O, okay, we know O is electronegative and it's more electronegative than hydrogen. So electrons are attracted, right? So electrons are going to spend their time near this electronegative element. So the oxygen, it becomes partial negative and the hydrogen will be partial positive, right? So that's why a permanent dipole is H2O. A water molecule is a permanent dipole. There's an uneven sharing of electrons, you can say, right? Okay, so when we talked about temporary dipoles, it's basically there's a temporary shift in the electron distribution. There might be they're closely packed, so nearby charge or maybe a dipole that already exists will cause this shift in electron distribution. And if there's like a polar molecule, a non-polar molecule might uh, it might become a dipole along with it. That's going into the physics, the distortion of the electron cloud happens. It's basically uneven distribution, and that's what you need to remember. And even electrons are always moving, so at some instant they're not evenly distributed. So the importance here is that we talked about the shape, right? They're holding the secondary tertiary structures by these dipole-dipole interactions. And the hydrogen bond that we mentioned, it itself is actually a dipole-dipole interaction. Okay. Another thing we mentioned here was asymmetric. It's also important to know what we mean when we say asymmetric. So asymmetric is basically that they are non-chiral. Okay. That if you mirror that structure, it's not going to be the exact same. Like if this is a structure, something, and you mirror it, right? And you keep it on top of each other, it's not going to uh, be superimposed. Superimposition. So this will not be superimposed. Just so you're sure we're talking about this, right? So because they have the different groups, right? And if you were to mirror it, you would see that they do not align in such a way that they are right on top of each other. So that's also very useful in the system, right? Because you want it to fit in a specific orientation, right? So if you want binding, you would want it to be in a specific orientation. So if you want a specific form of an amino acid, okay, we're familiar with L and D, right? That forms, these are actually sort of versions of each other that we know. So if we have an enzyme that targets this, it ensures it is not going to work with this one. Okay, so it basically means it's left-handed or right-handed. So it's different from its mirror image, and that is its functioning or how it interacts. A very important thing to know. Okay, the next point is actually relevant with what we just discussed. Stereospecific interactions. That the interaction between biomolecules is stereospecific. That this is what we discussed, that stereo actually means space. Okay, anything that refers to space uses stereo, and specific means only one way, right? In a selective way. So stereo here means that it's going to react to only one version of it, right? We know stereoisomer. What we were just discussing, L and D, these are isomers of each other, right? So that basically means that a biological process is going to happen only to one specific arrangement of atoms. Okay? To only it is stereo specific, so it will only recognize with one kind of stereo isomer, not the other one. Yeah. So we also understood the importance right before. A nice way to think about it is about how a glove only fits your right hand, but it wouldn't fit your left hand, right? Okay, let's finish all of the other points as well. We talked about these being carbon compounds, some features of some properties of carbon compounds. Okay, first of all, they can form covalent bonds, right? What are covalent bonds? Electron pair sharing, quite familiar with it. They can also form single bonds between two carbons. These are, by the way, very stable. Okay, they also have this ability, and they can also form double bonds. And these covalently bonded carbon atoms can then go on, and they can form linear chains, they can form branched chains, and they can form cyclic chains. You can understand by the name itself what kind of structure that would be, right? And we said that organic molecules have specific shapes and dimensions, right? So if we expand on that, it's just important to note a few things. Okay, first of all, they can be tetrahedrally arranged, right? The carbon atoms. And between two carbon atoms you see an angle of 109.5 degrees, and also there is a complete freedom of rotation around these C-C bonds. Okay, so around these bonds you also have a complete freedom of rotation. Okay, so कहीं पे भी align हो सकते हैं, you can align anywhere, unless you have a large group, you know, that interferes. Also the bond length, if you want to know, it's 0.154 nanometer for this and 0.134 nanometer for this. Okay, and we've already talked about the functional groups here, right? We may have missed aldehyde group is CHO. We also have thiol group, which is SH. Ester is RCOO, and this is ketone actually, right? That's it. So aldehyde is CHO. My bad, this is ketone. So that's it for today. Also, you should know that these functional groups are more reactive chemically than any of these hydrocarbon backbones, right? That's how they give them the chemical property, right? They're chemically more reactive. In the next one, we'll go into some definitions and terminologies that are very important to this, and then we'll talk about different conformations and structures. Basically, we'll cover over that. Moving on in the third video to fats and carbs in details.